In this next tutorial session, we're going to be looking at two further functions from the HE Toolkit that allow us to process our biology sample data into a format that makes it suitable for hydroecological analysis and modelling. Looking at our flowchart, the two functions that we're going to be focusing on here are the predict indices function and the join HE function. When we were earlier on in step one looking at how we could import macroinvertebrate data, you will remember that the data that we import from the Ecology Data Explorer gives us observed metric scores for a wide variety of community level metrics. However, if we want to use that data to assess the impact of different pressures on that invertebrate community, then we need to not only know what the invertebrate community looks like on the day it was sampled, but also have a reference point describing what the invertebrate community should look like in the absence of any anthropogenic pressures. Many of you will be familiar with the River Invertebrate Classification Tool, or RICT. RICT is a tool, a long-standing tool, that allows us to predict what the invertebrate community at any given sampling location would look like in the absence of those anthropogenic pressures, and therefore provides a reference point that we can use to measure the health of the invertebrate community and also to generate Water Framework Directive classifications. The RICT tool is publicly available, and it's available via the Microsoft Azure platform. This means that we have the facility to take environmental or physical data about our sampling location, feed it into the RICT tool, and then extract or get back expected scores for a wide variety of invertebrate indices. The RICT tool is fantastic, but all of that happens outside of the R environment, which means if we potentially have to export data from R, run RICT, and then import data back again. However, in recent years, the functionality of RICT has been mirrored in R using a new package called RICT. And to leverage the use of that new RICT package within the HE Toolkit, we've developed a wrapper function called predict indices, which calls the RICT predict function and allows us to calculate expected macroinvertebrate indices for one or more sites of interest. To run the RICT tool, we need the environmental base data for each sampling location. So that describes the physical characteristics of the site. As an absolute minimum, the RICT tool requires us to know the altitude and the slope of the site, and also to have substrate composition data. That is, we need to know the percentage composition of boulders, cobbles, pebbles, gravel, sand, and silt clay, and those four percentages must add up to 100%. This training workshop isn't the right forum to go into the technical details of how the RICT tool works, but if you're interested in understanding more about how RICT works and generates its predictions, then there's a link on the slide there that will take you through to the RICT technical specification and user guide. To run the predict indices function from the HE Toolkit is really quite straightforward. All we need to feed it is a table containing the environmental base data for all of the invertebrate sampling locations of interest. So this is the environmental base data that we generated using the import env function earlier. On the slide here, the table at the top shows an example data set. We've got environmental data for 9 different sampling locations. We feed that into the predict indices function, and the output is shown at the bottom of the slide here. So in the output table, we have one row per sampling location, and the predict indices function gives us those expected scores under reference conditions for a total of 80 different macroinvertebrate indices. Some of those are relatively obscure and you may never need to use them in practice, but in amongst those 80 indices are all the commonly used ones like WHPT N-taxa, ASPT, Life, and PSI. The function will also provide predicted probabilities for the different RIVPAX end groups, which provides an indication about what sort of invertebrate community we might expect to find at each sampling location. So the predict indices function really just gives us a very simple way, a quick and efficient way of generating expected values for our invertebrate indices from within the R environment. That gets us to a point where we have the observed metric scores from the import INV function. We now also have the expected scores from the predict indices function. And one thing which we might commonly want to do with that is to then calculate observed to expected ratios, or OE ratios. This is a relatively simple thing to achieve, and so we haven't written a dedicated function to do it. Instead, on screen, I've provided some example code just to illustrate how that process might work. The first step is to do a join. So here we've used a left join to take the biology data containing the observed scores, and then join in the predicted data containing the expected scores. And we join the data here on two common fields, the site ID and also the season, because the expected scores will vary slightly depending on whether it's a spring, summer or autumn sample. Having joined the data all together into a single data frame, then we can use a mutate function to, first of all, just rename some of the columns into a slightly more meaningful format, and then to calculate the OE ratios themselves by dividing the observed scores by the corresponding expected scores. This gives us OE ratios, or EQIs, which provide an indication of how healthy the invertebrate community is. An OE ratio at 1 or close to 1 indicates that the invertebrate community is pretty much in line with what we would expect if there were no anthropogenic pressures acting at that location. And ratios less than 1 indicate a progressively greater degree of anthropogenic impact upon the community. So that gets us to a point where we've got our invertebrate data processed into the form of OE ratios. And as we looked at in step 2, we also have a variety of hydrological summary statistics, which describes how the flow regime at each location has changed over time. The final step in our data processing journey is to bring all of that data together, to join together the disparate biology sample data and the hydrological or flow statistics into a single data frame that we can then use for hydroecological analysis or modelling. Whilst you can join the data sets together in a relatively simple way, there are quite a lot of nuances here, and there may be different requirements to join the data in different ways depending on how we want to analyse it. So to facilitate that process of joining the hydrological and the ecological data together, we've developed a function called join HE in the HE toolkit. This is designed to work seamlessly with the import INV function and also the calc flow stats function. Those functions generate the two primary inputs that we need to run this function. That is a biology data set containing macroinvertebrate data and a data set containing our flow statistics, which feeds it into the flow stats argument. One thing just to note, and you can see I've highlighted a few bits in the table here, is that this function expects those two data sets to contain certain hardwired column names. So the biology data must contain columns which are called biology site ID, that's a unique identifier for each site, and also a date column which indicates the date on which the sample was taken. Similarly, the flow statistics must sit in a data frame which has a flow site ID, indicating the location of that gauging station, and a start and end date, which define the time window for which the flow statistics have been calculated. If you use this function off the back of those earlier functions, then those column names will all be sort of ready, set up for you, so you shouldn't need to redefine anything. But just to be aware that if you're importing data from other sources, for example, you might need to manipulate some of the column names to match these hardwired column names that the join HE function expects. You can see also from the table here that there's a number of other arguments which give us quite a lot of control or flexibility about how we then join the biology and the flow statistics data sets. This is one of the more complicated functions in the HE toolkit. So on the next few slides, I'm just going to take a moment to talk you through each of those arguments in turn. First and the simplest of those arguments is the mapping argument. The mapping argument allows us to define how the biology sites should be paired up with the flow sites or gauging stations. We can do this by defining a simple lookup table, which incidentally forms part of the metadata table that we looked at earlier on in this course. On the screen here, we've got a very simple example. We've got 5 different biology sampling locations, and each of those is then paired up with a flow site ID, i.e. a nearby gauging station. If we are working with data where the biology and the flow site IDs actually are one and the same, they're identical, then we can actually skip this mapping argument altogether and just set it to null. The other thing to note is that a given flow site or gauging station can potentially be paired with more than one biology sampling site. Quite often we might be looking at data in a particular catchment. There may be a single gauging station at the catchment outlet, but 2 or more biology sampling locations within that catchment or water body. So it's entirely acceptable for 2 or more biology sites to be paired up with the same flow site. That just is a simple and pragmatic way of making the best use of the readily available flow data. So a mapping argument tells us basically which sites we want to join up with which other sites. The next step in the process is for us to tell the function exactly how we want to join the biology sample data with the flow statistics for different moving windows. You remember in step 2 we looked at the calc flow stats function and explored how you can use that to divide up a time series of daily flows using a moving window and to calculate flow statistics for each of those windows or time steps. When we're doing hydroecological analysis, we're interested in trying to understand the effect of antecedent flows on the biological indices that we calculate on each sampling occasion. And the schematic on the right here, I've got a sort of a simple diagram to try and illustrate this process and to show how the join HE function gives us some flexibility or choice about exactly which historical time periods we want to take flow statistics from. In this example, we've divided up our hydrological time series into monthly time steps. So we've got summary statistics, flow statistics for each month: June, July, August, September, and October. And in this particular example, we've got one invertebrate sample taken in the middle of September, and that's represented by the black cross here that hopefully you can see on the right-hand side of the screen. We start off by numbering the different time periods for which we have flow statistics. So lag 0 is either the most recently finished flow period. So in this case, if our invertebrate sample was taken in September, the most recently finished flow period would be August. So that would take a lag of 0. And then from that, we count back. So then July is lag 1 and June is lag 2. And we call that method rule A. Alternatively, we can define lag 0 to be the most recently started flow period, in which case September becomes lag 0, August is one time step before that, July is lag 2, June is lag 3. So the method argument basically sets the numbering of those different flow time periods. And then having numbered those, we can then tell the function which of those lagged flow periods we want to take the flow statistics from and join up with our biology data. This essentially gives us control about how far back in time we want to consider the antecedent flow record when trying to explain spatial and temporal patterns in our ecological data. The example I've just talked through at the top there uses contiguous flow periods, which is probably the most simple and straightforward way in which we could characterize our flow statistics. The second example lower on the slide shows exactly the same process, but this time we've got overlapping flow periods. So here we've got a 2-month moving window which steps on by 1 month each time. The process, however, works in exactly the same way. We define the method to be either rule A or rule B, and then that defines which time period is lag 0, and then subsequently we can count back from that to identify the time periods which are lag 1 and lag 2 and lag 3 and so on. We then, when it comes to actually joining the biology and the flow data, we can specify one or more different lagged time periods for which we want to take the summary statistics and join them up with our ecology data. If that sounds a little bit abstract, then let's look at a couple of simple worked examples to see how that works in practice. You'll notice here that we've got a fourth and final argument that we need to configure, which is the join type argument. Join type really just tells us whether we want to start with the biology data first and then join the flow statistics to it, or vice versa. So in this first instance, we set join type equals add flows. So that means we're going to start off with a table containing our biology sample data and then join the flow statistics to it. Just to illustrate this, I've got a really simple hypothetical data set. We've got a single biology sampling location, and we've got two separate sampling occasions, one in April and one in September, and each of those generates a metric score. For the flow data at that same location, we've defined a moving window of 1 month. So we have a series of monthly flow statistics, and each month we've calculated a Q95, standardized Q95 statistic, which you can see in the right-hand column here. The code on the left shows how we're going to use the join HE function. We bring in the biology data and the table of flow statistics as our two primary inputs. In this example, the biology site ID and the flow site ID are identical, so mapping can be null. We don't need to specify a link table. We're using method A. So here we want to take the biology data and join in the flow statistics for the immediately preceding, the most recently finished time period, that is lag 0. Lags, we're only interested in that lag 0 time period, but we could join in flow statistics for other lagged periods if we wanted to, just by expanding out the figures inside the concatenation here. So for example, we could have 0, 1, 2 if we wanted the flow statistics from lags 0, 1 and 2. And then join type is add flows. So we take our biology data and we join in the flow statistics. So the box here shown with the blue outline shows the output from this function. So we have the biology sample data in the first 3 rows, and we have the flow site ID, we have the window number and the start and end dates for the lag 0 period, and then we have the Q95 statistic joined in for that lag 0 time period. You can see the output table has the same number of rows as our input biology data set. So we have 2 samples in that data set, so we have 2 records in our output table. Our first biology sample was taken in April, and that's joined up with the lag 0 time period, which is the most recently completed time period. So that's time window 3, which, as you can see, is March. That's the most recently completed time period. And the Q95 value is 0.97. And similarly, the September sample is joined up with the August flow statistics. So here we're describing the antecedent flow conditions in the 1 month period immediately prior to the month in which the sample was taken. Joining the data in this way, starting with the biology data, then joining in the flow statistics, is the approach that we would take if we wanted to build a data set, a calibration data set, for then building a statistical model. It ensures that we have a data set which contains a response variable, a biology metric, and then 1 or more predictive variables. Those are our flow statistics. There are instances, however, where we want to do the process the other way around. We want to start off with a table of flow statistics and then join in the biology sample data for those time periods when we have biology records available. So here we've got exactly the same example data set. We've got 2 biology samples, and a table of monthly flow statistics. The code we use is exactly the same as we had on the previous slide. The only difference now is that join type is set to be add biology. So we're starting with the flow statistics and then we're adding in the biology records. Again, we're using method A and lag 0. So we're joining the two data sets to identify the flow statistics from the most recently completed month prior to when each biology sample was taken. The table at the bottom, highlighted with the blue box around it, shows us the output. Instead of just having two records now, we have the same number of records as we had in our table of flow statistics that fed into the function. We have a table of flow statistics, a sequence of window numbers from 1 to 12, corresponding to January to December. And you can see that the biology data for April is joined to the flow statistics for March, and the biology data for September is joined to the flow statistics for August. So again, this table shows us what the flows were like in the month prior to when each biology sample was taken. Joining the data this way gives us a data set we can use either to create some time series plots to explore how the flows and the biology data have changed over time at each location. And formatting the data or joining the data in this way is also useful because it allows us to use the range of flows function, which allows us to see how well the biology data cover the full range of hydrological variability that we've observed historically at each location. The range of flows function we're going to cover in workshop 2, but the time series plots, the HEV plots, we're going to look at in our next tutorial. So there we've covered the use of two functions which allow us to process our invertebrate data and then join our biology data, be that invertebrate data or some other biological receptor, with our flow statistics to generate a data set for modelling or for further data visualization. If you'd like to have a go at using those two functions, then again, I'd refer you over to the POSIT Cloud project we've set up. Open up Practical Exercise 3, and that contains some example code that will allow you to run both of those functions and explore how changing some of those arguments affects the output from those functions.